Hello students, today I am here for discussion of previous year question. So the first question I am going to discuss with you is v-SNARE and t-SNARE, and it is a snare hypothesis and it is a repeat question. v-SNARE and t-SNARE, this topic is from Golgi complex. Means this is found in Golgi complex and involved in vesicle fusion. So what is the full form of v-SNARE and t-SNARE? v-SNARE and t-SNARE, the full form of snare is soluble in ethylmalamide sensitive factor activating protein receptor, and V for vesicle and T for target membrane. So this snare is nothing but SNAP receptor, and the full form of SNAP is soluble NSF attachment protein, and full form of NSF is NEM sensitive factor. Now let's discuss in detail about this snare, v-SNARE, t-SNARE and snare hypothesis. So as I have already discussed with you that this is found in Golgi complex. Golgi complex is a organelle according to your syllabus, and this is involved in vesicle fusion. 4 parts of Golgi complex are cisternae, tubules, vesicles and vacuoles. And vesicles are membrane bound sacs that store and transport modified proteins and lipids within a cell. So what is vesicle? Vesicle is a membrane bound sac, and this vesicle store and transport modified proteins and lipid within a cell. So the main function of this vesicle is store and transport modified protein and lipid within a cell. Now what is vesicle fusion? And this snare helps and involved in vesicle fusion. So what's about vesicle fusion? Vesicle fusion means transport vesicle found, and fusion, vesicle fusions means fusion between transport vesicle with its target. Okay, so 2 types of event occurs during this vesicle fusion process. First of all, transport vesicles must specifically recognize the correct target membrane. First step is recognition. Recognition of what? This transport vesicle must specifically recognize the correct target membrane. Why this recognition is very much important? Because this transport vesicle and target membrane is going to fuse. Okay. Then the second step is fusion of vesicle and target membrane. So 2 types of event occurs here. First of all, recognition of transport vesicle and its correct target membrane, and the second step is fusion of vesicle and target membrane, and this event is mediated by specific recognition between a vesicle and its target by interaction between unique pairs of transmembrane protein, and this is mediated by specific recognition between a vesicle and its target by interaction between unique pairs of transmembrane proteins. Transmembrane means this protein is some part of this protein inside plasma membrane, and some part of this protein is outside of plasma membrane. So transmembrane. Okay. Then the next step is fusion between phospholipid bilayer. phospholipid bilayer. All of we know that phospholipid bilayer is a character of plasma membrane, protein lipid protein membrane, and it is known as phospholipid membrane and phospholipid or phospholipid bilayer. So the next part is fusion between phospholipid bilayers of the vesicle and target membranes. To clearly describe this phenomenon, a hypothesis was proposed. Hypothesis, the name of that hypothesis is SNARE hypothesis, to explain vesicle fusion, and this SNARE hypothesis model was proposed by Rothman and his colleagues, scientist Rothman and his colleagues. According to this model, what happens? Vesicle fusion with target is mediated by interaction between specific pairs of protein called SNAREs. Okay, full form of SNARE we have already known, that SNAP receptor on the vesicle v-SNARE, as we have already discussed here, v-SNARE, V for vesicle and T for target membrane. So, vesicle fusion with target is mediated by interaction between specific pairs of protein called SNAREs, and on the vesicle it is v-SNARE and target membrane it is t-SNARE. V-SNAREs integral proteins pairs with cognate t-SNAREs in target membrane. In neurons, for example, in neurons, v-SNARE present on synaptic vesicle is designated as synaptobrevin, and t-SNARE are designated syntaxin, and SNAP25. Okay, so the first is recognition step. This step is controlled by members of a family of monomeric GTPase called RAB protein. GTPase protein, RAB protein. GTPase means this protein helps to break GTP and GDP. Okay, GTP means triphosphate, 3 phosphates are there, and GDP means diphosphate. Okay, so one inorganic phosphate will be released with energy. Okay, so recognition step is controlled by members of a family of monomeric GTPase called RAB protein. So RAB protein is very much necessary for this recognition. Okay, which check what the RAB protein function. What is the function of RAB protein? This RAB protein check that the interaction between a v-SNARE and a t-SNARE, if it is correct or not. Okay, RAB proteins make attachment with the surface of vesicle. Okay, when a vesicle encounters the correct target membrane, the binding of v-SNARE to t-SNARE causes the vesicle to remain bound for long enough to allow the RAB protein to hydrolyze its bound GTP, which locks the vesicle onto the target membrane. Cells contain many types of RAB proteins, each associated with the membrane of particular organelle involved in the secretory or endocytic pathway. After the v-SNARE and t-SNAREs have mediated the fusion of a vesicle on a target membrane, the NAC, full form of NAC, this is NEM sensitive factor, binds to SNARE complex via adapter protein SNAP. SNAP full form soluble NSF attachment protein. NSF is soluble to ATPase and hydrolyzes ATP to dissociate the SNARE apart. Okay, concentrate in this figure, and if it is broad, if the number will carry 3 marks, then you should explain it with diagram, proper figure, proper diagram. Now let's concentrate. This is vesicle. Okay, this is vesicle. Vesicle means vesicle. All of we know vesicle means Golgi complex vesicle. Okay, vesicle of Golgi complex, and here RAB GTP is attached. Okay, and v-SNARE, vesicle snare protein is attached. It is normal position. Now the next step, here look, this is target membrane, and the target of this vesicle is to bind with this target membrane. Okay, now the next step, here look, t-SNARE. T-SNARE, full form of t-SNARE is which? What? It is t-SNARE means target membrane snare protein, and the first step of vesicle docking is occur. So vesicle come in close contact with target membrane t-SNARE. This is v-SNARE and this is t-SNARE. And look, the next step, formation of snare complex. How the formation of snare complex occurred? v-SNARE and t-SNARE attachment. Okay, and the docking occurs. Okay, docking occurs. Now till now this RAB GTP is remain intact. Okay, the next step, and the formation of snare complex occurred. The next step is GTP hydrolysis, and this RAB GTP is now, after hydrolysis, it becomes soluble RAB GDP. Okay, diphosphate, triphosphate to diphosphate. Then this vesicle and membrane fusion occurs. Look here, the vesicle and membrane fusion occurs, and this v-SNARE and t-SNARE complex is here. It is no longer attached with this vesicle and fusion membrane. Vesicle and membrane and fusion occurs. Okay, then adapter proteins appear, and NSF and ATP is being attached, and the next step is snare dissociation. The v-SNARE and t-SNAREs are disassembly of snare complexes occurs. This v-SNARE and t-SNARE now is separated. Okay, so this is the figure description. This is the figure describing complementary sets of vesicle snare means v-SNARE and target membrane snares t-SNAREs. Okay, determine the selectivity of transport vesicle docking. V-SNAREs of transport vesicles from the donor membrane bind to complementary t-SNAREs in the target membrane. The RAB protein hydrolyzes its bound GDP, locking the vesicle onto the target membrane and releasing RAB GDP into the cytosol. From there, it can be reused in a new round of transport. The vesicle then fuses with the target membrane. NSF is an ATPase. NSF means here NSF. Okay, NSF. Okay, NSF is an ATPase that hydrolyzes ATP to release the complex once it has done its job. Okay. Here look, NSF plus ATP. NSF and ATP, adapter proteins, NSF and ATP. NSF is ATPase. Okay, ATPase means NSF is able to break this ATP into ADP plus inorganic phosphate by hydrolyzing this ATP. Okay, and to release the complex once has done, and it helps to release this complex once it has done its job. So it is in detail the v-SNARE and t-SNARE. Okay, and snare hypothesis of Golgi complex. The next session is coming soon.